Hello everyone, and this is Ashutosh, the TA for Environmental Biotechnology. So today is Saturday 3 p.m. and it's already 3:05. We are going to start the class. So I welcome everyone to this class. Good evening, Ashwini. I welcome everyone to this live TA session and problem solving series for week 8. We are in the 8th week of Environmental Biotechnology, and today we'll be dealing with all the lectures taught in week 8, basically bioremediation. This course is taught by Professor Pinaki Sar from IIT Kharagpur, and my name is Ashutosh. I'm from IIT Kanpur, a PMRF PhD scholar. So, for your information, this class is a recorded class, and the recordings of this class will be uploaded on YouTube, and the link will be provided to you by the NPTEL authorities. The materials that I will be sharing in this class will also be provided to you in the form of a PowerPoint presentation. So today in today's class I have designed 10 such questions corresponding to lecture week 8, in which we will see some of the example assignment questions very similar to what you will face in your assignment, multiple choice questions in which one of the options is correct, and then briefly I will explain the concepts and the definitions and the logic behind the correct answer. So this is the format that I am going to follow throughout this course as well as in every live TA session. I will post a PowerPoint presentation on the screen with multiple choice questions one by one. I will ask the students present in the live class as well as in the recorded class to please post your answers in the chat box, and after a minute or two I will go through all the answers and reveal what is the correct answer, and then we will together understand what is the concept behind the correct answer. Okay, so this is a general instruction for all students who want to join this class, and anyone who is requesting to join this link will be allowed to do so. And I would request all the participants in the live class to please turn off your mic and video chat for smooth conduction of this session. You can communicate your answers and as well as talk to me in the chat box, which I'll be following regularly. Okay, without any further delay, let us start this session. Let me present my screen. Yeah, I hope everyone can see my presentation and see the questions and slides changing, and my audio is clear to everyone. Then please let me know in the chat box with just a yes or a thumbs up, and then we can begin this. Very good. Okay, so once again I welcome everyone to Environmental Biotechnology Week 8. This course is brought to you by NPTEL and taught by Professor Pinaki Sar, and my name is Ashutosh. I'm I'm a PhD scholar at IIT Kanpur, who is the teaching assistant for this course. Okay, so this is, again I'll repeat, this is the format of the materials and the presentation that we are going to have in this 2 hour long live session. I have prepared multiple choice questions which are similar to what you will be facing in your exams or in your assignments, and this will help you to understand the concepts behind such questions and revise your topics whatever you have read through the lecture series by Professor Pinaki Sar, and will also adapt you to the way of answering these questions when you actually face them in the exam. Okay, so without any further delay, let us look at question number 1 of this session. Which one of the following options presents the correct order of steps involved in whole metagenome analysis? Understood the question? Which one of the following options present correct order of steps involved in whole metagenome analysis? Option A, DNA sequencing, DNA fragmentation, assembly, phylogenetic binning. Option B, DNA sequencing, assembly, phylogenetic binning, DNA fragmentation. Option C, DNA fragmentation, DNA sequencing, assembly, phylogenetic binning. Option D, DNA fragmentation, DNA sequencing, phylogenetic binning, assembly. Or option E, assembly, phylogenetic binning, DNA fragmentation, DNA sequencing. And from left to right is from start to finish of a sequence. You have to tell me what is the correct order of steps involved in metagenomic analysis. Okay, so let us take a minute for all those who have attended the live class as well as who are going to watch this recorded session. Please post your answers in the chat box or the comment box for me to check. Let's take a minute or so to collect some answers, and I will be back to this question to tell you what is the correct answer to this question. I would encourage all the participants to participate in discussion, in answering, which will boost your confidence as well as you can gain the maximum out of such live TA sessions. I think it's enough time for this question. Time to check. Time to check for the answers. Ashwini has answered option D. Okay. Now let us see what is the correct answer to this question. The correct order of steps involved in whole metagenome analysis. Correct answer is option C. So first the DNA is fragmented, then the DNA is sequenced, and then comes assembly of those fragments into a whole genome with referencing, and then comes phylogenetic binning. Sorry Ashwini, you got confused between last two steps. Fragmentation and sequencing is correct, but assembly happens first and then phylogenetic binning. Okay, so what happens in the metagenomic analysis? Let us go step by step. So here is a sample full of cells, might be soil sample or a contaminated water sample, which has to undergo metagenomic analysis. The first step is DNA extraction. The samples are lysed and the DNA is extracted, as you can see. Then all the fragments are subjected to sequencing. So after all the fragments are subjected to sequencing, it will generate lot of reads, and these reads are now called contigs because they are contiguous elements of a whole single genome. This is first assembled into a meaningful organism's genome, and then many such organisms, because the sample consists of many such organisms, all the DNA will be binned together after generating a meaningful contig. So once the binning is done, then we can differentiate between different species and genes present in the community or in the sample. So this is the correct option. C is the correct one. First it is DNA fragmentation, DNA extraction or fragmentation, then it is sequencing, then the sequence are assembled into contigs or genome of the organism with the help of a reference genome, And once we have the reference genome for and contigs for all organisms, we use binning method with bioinformatics tools to identify different species and genes of this organism. What are the community composition of this? Okay, so what is the general workflow again? Putting it in words: DNA fragmentation. The extracted DNA from microbial community is broken into smaller fragments for sequencing. Then comes DNA sequencing. These fragments are then sequenced using high throughput sequencing platform like Illumina sequencing or PacBio. Then is the assembly step. Sequence reads are assembled into longer contiguous sequence which are called contigs. Then comes phylogenetic binning. The assembled contigs are grouped or binned based on a sequence similarity to assign what are the different taxonomical groups that are present in the community, in the sample. I hope it is clear to the students. First it is DNA fragmentation, then comes DNA sequencing, then comes assembly, and then comes phylogenetic binning. If yes, just let me know in the chat box and we can proceed ahead. Yes, very good. Coming back to question number 2, the second question of this session. Whole metagenome based analysis provides information about dashed. Whole metagenome based analysis provides information about dashed. A, option A, all organisms. Option B, all microorganisms. Option C, all prokaryotes. Option D, all bacteria. Option E, all bacterial chromosomes. A whole metagenome analysis. Which of these information is earned by a whole metagenome analysis? Let us take a minute and arrive at the right answer. Please post your answers in the chat box or in the comment box. Okay, I think it's time to check for answers. Ashwini says option B. Let us find out what is the correct answer to this question. Whole metagenome based analysis provides information about. Correct answer is option A, all microorganisms. Because it's a whole metagenome analysis. I think Ashwini got a bit confused again, but no issues. When you are doing a whole metagenome based analysis, not just microorganism, any organism present in the sample or whatever. So you need to understand that it is not specified whether it is a water sample or a soil sample, and there can be other organism other than microbes like bacteria or protozoans. If there are any multicellular organism as well, they will also be identified in a whole metagenome based analysis. So what is whole metagenome based analysis? Whole metagenome sequencing involves sequencing all the genetic material in an environmental sample. So unlike only selecting for 16S rRNA or filtering out microbial sample, it will sequence the all genetic material present in the sample. does not create any bias. So unlike 16S rRNA sequencing, which only captures bacteria, archaea, or metagenomics includes all microorganisms present, effectively bacteria, archaea, viruses, fungi, and other eukaryotes which might also be present. So it is not only limited to prokaryotes but many other organisms as well. So here is a feature-wise comparison between 16S rRNA sequencing and whole metagenomic sequencing. So what is the scope of 16S rRNA? If you compare the scope, bacteria and archaea only, while in whole metagenome sequencing, it is all microorganisms, bacteria, archaea, virus, fungi. Now the target of sequencing is 16S rRNA gene in 16S rRNA sequencing, while in whole genome analysis, whole metagenome sequencing, the entire genome is sequenced. Now what is the resolution? The resolution can differentiate between genus and species, and it is limited because it can only differentiate between bacteria, right? Whereas here the entire genome is sequenced, so we can now differentiate between species level and strain level as well. Function only on taxonomy, not much function, but for whole metagenome sequencing we can obtain taxonomy as well as functional genes because the whole genome is getting sequenced. Now of course the whole metagenome sequencing has a high cost because it also screens for all the functional genes. And what is the use of such a sequencing based method? For knowing the community composition or diversity surveys, 16S rRNA sequencing is enough, but for whole metagenome sequencing, taxonomy, functional, novel gene discovery, a whole metagenome sequencing has to be used. Now here is a workflow of comparison between 16S rRNA and whole metagenome sequencing. In whole metagenome sequencing, the reference microbial genomes, which is obtained after fragmentation and sequencing, which we saw in the previous question, can be used for these purposes. It can be used for high resolution and comprehensive taxonomic and functional profiling as well. Discovery of novel microorganisms like bacteria, fungus, and viruses, since we are sequencing the whole genome in the sample. Also discovery of novel proteins and tracking interaction between bacteria and transmission, study of evolution of the microbiome present in the sample, and metabolic modeling of personal microbiome if it is from a patient sample or somewhere else. So the questions are little tricky and can lead to confusion, so I am here to guide you through that. So I hope you understood what is the difference between 16S rRNA sequencing and a whole metagenome sequencing. Therefore, the answer will be option A, all organisms present in the sample. I hope it is clear to the students. If yes, then let's move ahead. Please let me know in the chat box. Thank you. Very good. Moving on to the third question. Third question is about bioremediation. Bioremediation was first applied to treat dashed. The first application of bioremediation, option A, oil contaminated water, option B, plastic pollution, option C, municipal wastewater, option D, industrial wastewater, option E, heavy metal contamination. Now which of these is an age-old practice or first application of bioremediation techniques? Is it oil spills, plastic pollution, municipal wastewater, industrial treatment, or heavy metal contamination? If you understood the question, let's take a minute and collect some answers, and I will return back to this question to reveal what is the correct answer. Bioremediation. I think it's enough time for this question. Let us check some answers in the chat box. Ashwini has answered option A. Let us see what is the correct answer. Bioremediation was first applied to treat. Correct answer is option C, municipal wastewater. I know again it is a confusing options. These are confusing options. Oil contaminated water and municipal wastewater are the two closest answer, but the correct answer will be option C, municipal wastewater. Now let us understand what is bioremediation and how municipal wastewater treatment is considered the first application of bioremediation. Bioremediation, definition wise, means using living organisms, usually microorganisms or some plants, to detoxify, degrade, or remove environmental pollutants. So, these are the environmental pollutants, might be plastics or garbage or organic matter, using plants, fungus, or bacteria living organisms to detoxify or degrade and remove these environmental pollutants to give back the green earth. And what are the advantages of bioremediation? It is totally environmentally friendly and natural process and very cost effective compared to physical or chemical methods like incineration or landfillings. Now if you remember, wastewater treatment is carried out by activated sludge process, which was developed around 1914 to 1916 in the UK. It's a wastewater treatment method where aerobic microbes are cultivated in aeration tank. These microbes consume organic pollutants, converting them into CO2, water, and new biomass. The microbial sludge is then separated and partly recycled back into the aeration tank to maintain a healthy microbial population. So this is a method of biodegradation which will come, bioremediation, which we will come across as engineered processes or activated sludge, which is an XC2 process, where the activated sludge or already biodegradable, the organic pollutant degrading bacteria culture is added onto fresh sewage water for municipal treatment. Thus, activated sludge can indeed be considered the first large-scale systemic application of bioremediation. It predates modern bioremediation projects like oil spill cleanup by decades, making it a foundational example of using microbes for environmental cleanup. So, by the traditional definition of bioremediation, the sewage treatment or municipal waste treatment by activated sludge process is an example of bioremediation, and it is an age-old practice as old as 1900s. While oil spills, the major application of microbe for bioremediation of oil spill was in the Alaskan oil spill, Vaders Exhaust Vaders oil spill, and other areas. So which is pretty recent in 1960s to 80s. This is 50 years earlier than that, which is activated sludge process used in treatment of municipal and sewage sludge water. So that is why option C is the correct answer. It can be confusing between option A and option C. I hope the doubts are all cleared. Okay, thanks Ashwini. Let's look at the fourth question in our session today. This is question number 4. Which of the following statements about bioremediation is true? Option A, bioremediation always requires human intervention. Option B, bioremediation cannot completely eliminate pollutants. Option C, microorganisms, pollutants are always toxins. Option D, bioremediation may also be performed by insects and plants. Option E, bioremediation always involves total elimination of toxicity. Which of the following statements about bioremediation is true? I hope you have understood the question. You have to find the true statement among these five options and post your answers in the chat box. I will return back to this question in a minute. Okay, let's come back to this question and check the comment section for answers. Ashwini has answered option D. Okay, and I hope everyone in the live session, recorded sessions, are also answering in the comment box. So, what is the correct answer to this question? Which of the following statements about bioremediation is true? Correct answer is option D. Bioremediation may also be performed by insects and plants. Correct one, Ashwini. Well done. So let us understand why D is the correct answer, which is a true statement about bioremediation. So let us understand about the other options. Bioremediation cannot completely eliminate pollutants. Pollutants are always toxins and always requires human intervention. Right? So one of these options tell us that bioremediation removes toxicity completely. So this is not true because microbes cannot completely degrade many pollutants. Example, plastics, heavy metals and complex synthetic chemicals. So it cannot completely eliminate pollutants. But there are some pollutants which it can completely eliminate, like hydrocarbons, like petroleum products, which can be utilized by hydrocarbon as an energy source to break down into contaminants into harmless substances. So this negates option B and option E completely. Total elimination of toxicity, not true, and cannot completely eliminate pollutants, which is partly true, because some pollutants which are cannot be completely eliminated, while there are some pollutants like hydrocarbons or petroleum which can be broken down into harmless substances such as carbon dioxide and water during bioremediation, because microbes use them as the source of their energy. So converting all these contaminants or pollutants into non-toxic metabolites is the maximum they can do. Bioremediation often reduces toxicity rather than removing the contaminant entirely. This is also true, guys. You have to notice that bioremediation does not aims on removing the contaminant totally. It can only reduce the toxicity from a non-toxic metabolite. So in many practical cases, cleanup is incomplete. So here is another example of oil spill where the entire contaminant is removed, because hydrocarbons in the oil can now be utilized by the hydrocarbon degrading bacteria to produce H2O and CO2 in return, and production of CO2 and H2O and oxidation of the hydrocarbon, because the bacteria is consuming the hydrocarbon as a source of its energy, thereby degrading the petroleum oil spill. Now looking at the correct option, which is option D, insects and plants which are also used in bioremediation. When insects are used for bioremediation, it is known as entomological bioremediation, which is insect based remediation. Use of insects or insect DI process to help degrade, transform, or remove environmental pollution or waste. And various famous insects include wax moths, soldier flies, beetles, ants, and termites that can break down and absorb toxic substances, offering low energy method after environmental cleanup. So here is an example of the plastic eating, wax eating moth. Degradation initiates when the wax eating moth lays eggs onto the plastic. The caterpillar degrades the plastic with salivary enzymes, and gut enzymes and microorganisms digest it when it is ingested further inside. And then the caterpillar excretes certain droppings which consist of degraded products like oxidized aliphatic chains, which are no longer harmful. So such as an example of the wax eating moth, which is an entomological bioremediation based method. Now what is plant based bioremediation method? This is also known as phytoremediation. Phyto is light and remediation is the use of green plants to remove, degrade, or stabilize pollutants from soil, water, or air. For example, sunflower removes heavy metals like lead, arsenic, uranium from soil and water. Indian mustard, effective for extracting lead, selenium, and cadmium. And water hyacinth absorbs heavy metals and organic waste from wastewater. So these are some of the well-known examples of plants being used for phytoremediation. So plants can do it in various methods: volatilization, conversion of metals and metalloids to less toxic and volatile forms so that they can be soluble in air. They can be released in the air. Metals and metalloids are taken up by the root for uptake, manipulating metals, metalloids, transporter genes and uptake system, influx and efflux rates, detoxification in the leaf, translocation through the phloem and xylem. And enhancing metals and metalloid ligand production with all these capabilities. So plants are also widely used in phytoremediation, and here are some of the examples. That is why option D is the correct answer. Insects and plants can also be used for bioremediation. I hope it is clear to the students. Just let me know in the chat box and we can move ahead to the next question. If you have any queries or questions for me, please stop me and put it in the chat box. I will be happy to answer. If not right now, right here, then in the next class, of course. If you have some question, if I'm not able to answer right now. Okay, let us move on to the next question swiftly. Question number 5. Again, this is an exam question for bioremediation. A pond is contaminated with industrial wastewater containing dyes. Understood? Pond is contaminated with dye water from industry to treat this, dye degrading bacteria were added to the pond water. This is an example of dashed. Option A, in situ intrinsic process. Option B, ex situ instinct process. Option C, in situ engineered process. And option D, ex situ engineered process. Which of the following option would be correct answer for this question? Industrial wastewater containing dyes and you add a dye degrading bacteria. Into the pond water. Understood the question? Then post your answers in the chat box. Let us take a minute together and then I'll be back to the question for revealing what is the correct answer. Is it option A, option B, C or D? Post it in the chat box. Okay, I think it is enough time for this question. Let us see what is the correct answer to this question. A pond is contaminated with industrial wastewater containing dyes. To treat this, a dye degrading bacteria were added to the pond water. Let me check the chat box if there are some answers. Ashwini has answered option C, Shailaja option A, and Ramya has answered option C. Let us see what is the correct answer. Option C is the correct answer. So this is a process of in situ engineered process. In situ because the pond and the sample is still there in its native habitat, and an engineered process because dye degrading bacteria was added into the pond water. So it's an engineered process. Okay, so Ashwini and Ramya, good answers. Your answers are correct. Sailaja, maybe better luck next time. Okay, let us understand what is the difference between in situ and ex situ and intrinsic and engineered processes. So in situ bioremediation, as the name suggests, treatments of pollutant at the site of contamination without excavation or removal. So if you do not remove or excavate the material, you treat the pollutants at the site of contamination. This is in situ bioremediation. In situ means inside the place. Now ex situ bioremediation, contaminated soil, sediment or water is removed from the site, treated somewhere else in bioreactors or at treatment facilities, and then got brought back into the site. So this is ex situ bioremediation, removing the indigenous material or native specimen from a site, treating it somewhere else and getting it back. So feature wise, if we compare the in situ bioremediation and ex situ bioremediation, the most important difference is the location. In situ is on site while ex situ is off site, mostly excavation. In cost, in situ bioremediation is cheaper, whereas ex situ is more expensive. In situ is slower, while ex situ is faster. The control over in situ bioremediation is limited because it is site dependent, while for ex situ it is higher control because the conditions are laboratory controlled or reactor controlled. Minimal disruption to the native environment in situ, while excavation disturbance required for ex situ. Sorry. And what are the examples of in situ bioremediation? Bioventing or biosparging, while ex situ is biopiles and slurry reactors composting. Okay, so these are the point of differences between in situ bioremediation and ex situ bioremediation. Now, what is the difference between intrinsic and engineered processes? Intrinsic bioremediation or natural attenuation relies on the natural microbial population already present in the contaminated site. So, in the contaminated site, in our example here, the pond containing the dye water, there are already some naturally occurring microbes which are present in the contaminated site. If those can be used for bioremediation, then that is a type of intrinsic bioremediation. No external stimulation required, no nutrients, no oxygen, no extra microbes added. So in intrinsic method, there is no external stimulation or microbial addition. Pollutants degrade slowly but naturally under existing environmental conditions. So this is a slow method and often monitored but not actively enhanced. So there is no human intervention as well. For example, natural breakdown of spilt petroleum hydrocarbons in soil by the indigenous microbes present in the soil, natural degradation of sewers in river by native microbial communities. These are an example of intrinsic bioremediation. Now, what is extrinsic bioremediation? Human intervention is used to stimulate microactivity and speed up the process. Yeah, here this process involves adding nutrients like biostimulation when nutrients are added, supplying oxygen or air like bioventing or biosparging. This is what we saw. Introducing specialized microbes like bioaugmentation. So these are methods of extrinsic or engineered bioremediation. This is more controlled, faster and efficient compared to the intrinsic methods. For example, pumping oxygen and nutrients into oil contaminated soil to accelerate its degradation, or adding engineered strains for enhanced petroleum hydrocarbon degradation. So thereby the dye problem in the pond, where it is in situ because the pond is still situated there and the remediation is done inside the pond, and an engineered process because dye degrading bacteria, engineered or extrinsic process of bioaugmentation where specialized microbes are introduced. So this is an example of bioaugmentation, extrinsic or engineered in situ process of bioremediation. I hope it is clear to the students. If you have any doubt, please let me know. I can repeat this as many times as you want, but I want everyone to be clear on the concepts. Ashwini says okay. Okay. If it is clear, then I would like to move forward with question number 6. Again, a problem related to bioremediation. Plastic waste at a landfill was found to harbor a population of bacteria naturally capable of breaking it down. Addition of nitrogen rich organic waste was observed to accelerate this process. This is an example of dashed. Option A, biostimulation. Option B, bioaugmentation. Option C, composting. Option D, bioreactor. Option E, Land farming. I hope you understood the question. There is a plastic waste at a landfill which has naturally occurring bacteria that can break the plastic. But it is observed that after adding nitrogen rich organic waste, this process is accelerated. This is an example of which method? Option A, biostimulation. Option B, bioaugmentation. Option C, composting. Is it a bioreactor or land farming? Okay, let us take a minute or so and arrive at some answers in the chat box. I will come back to this question and tell you what is the correct answer. Okay, I think it's enough time for question number 6. Let me check what are the answers in the chat box. Only Ashwini has answered option A. Okay. Let us see what is the correct answer to this question. Correct answer to this question is option A, biostimulation. Well done, Ashwini. So this method is a biostimulation. This is a process of in situ engineered process. Again, so what is the difference between biostimulation and bioaugmentation? In the previous example, in the previous question, we saw bioaugmentation. Biostimulation, addition of water or nutrients or any such material which can help indigenous microorganisms to remove contaminants better. This is biostimulation, stimulating the already present indigenous microorganisms. What is bioaugmentation? Augmentation means support. So additional microorganisms are supplied into the site for easy removal of the contaminant. So we have added this yellow microbe which is engineered to degrade this contaminant along with the indigenous microorganisms. So this is bioaugmentation. This is the major difference. Addition of live microorganisms for bioaugmentation, while addition of nutrients and waters or media which will help the indigenous microorganism to remove the contamination. And at the end we receive the successfully bioremediation of the sample. Now, since in the question the microbes were already present and capable of plastic degradation, by adding nitrogen rich waste, their activity was enhanced, which is a classic definition of biostimulation, stimulating the already present plastic degrading bacteria with nitrogen rich waste. So bioremediation can be classified majorly into in situ and ex situ bioremediation. Here I will explain all the types present in the bioremediation topic. In situ bioremediation on the site itself, ex situ excavated from the site. In situ can be further classified into two types: intrinsic bioremediation and engineered bioremediation. Biosparging, bioventing, bioslurping, biostimulation is an engineered bioremediation because we are adding nutrients from the external sources. Bioaugmentation, we are adding microorganism from external sources, and phytoremediation. All these are in situ engineered bioremediation techniques. In ex situ bioremediation, we have slurry phase bioremediation, which is a bioreactor like the activated sludge, and the solid phase bioremediation, biopiling, land farming, biofilter, and composting. Now, explaining everything in detail, in situ bioremediation again, treatment is done at the contaminated site without excavation. Natural and intrinsic bioremediation, natural microbial process degrade pollutants without intervention of humans, such as natural breakdown of hydrocarbons in soil. Engineered or enhanced bioremediation, human intervention is required to accelerate the microbial activity, like biosparging, injecting air or nutrients into the groundwater to stimulate the microbes present in the deeper layers of the soil. Bioventing, supplying oxygen to contaminated soils for aerobic bacteria mediated degradation of contaminants. Bioslurping, using vacuum pumps to remove and treat hydrocarbons which are spilled into the soil. Biostimulation, by adding these nutrients, which is an example where we stumbled upon this question itself, to boost the native microbes present around. Augmentation, adding specialized microbial strains. And phytoremediation, using plants to remove or stabilize or degrade pollutants. Ex situ bioremediation, contaminated soil or water is removed from the site and treated elsewhere. Excavation is needed. Slurry phase bioremediation is a bioreactor, soil mixed with water and treated in a bioreactor. This is faster but is expensive. And solid phase bioremediation, contaminated material treated in solid form. There is no slurry or water involved. There is no solution involved. Biopiling, like piling contaminated soil with aeration and nutrients. Land farming, spreading soil on land and tilling to enhance microbial degradation. Biofilter, passing contaminated gases or liquids through microbial filters. And composting, mixing contaminated material with organic matter and earthworms to stimulate microbial activity. So these are all the classifications of types of remediation, bioremediation. And the correct answer to this question was biostimulation, in which plastic degrading bacteria already present on the site is stimulated by addition of nutrients like nitrogen rich waste. I hope everyone understood these concepts because these are very important. Bioremediation concepts. Let me know in the chat box if you want me to repeat any topic. If not and you have understood everything, then please tell me a yes or no and we will move ahead in the question. Okay, very good. It's good that you are getting the concepts and understanding it. Okay, thanks Ashwini. And moving on to the next question. Question number 7. One of the oldest bioremediation systems is the activated sludge system. Here, a fraction of solid waste containing microorganisms and partially treated waste is added as to new untreated waste in a wastewater treatment plant. This is best described as dashed. Again, a classification based question. Activated sludge system in which a fraction of solid waste containing microbe and partially treated waste is added to new untreated waste in a wastewater treatment plant. What kind of bioremediation process is this? Option A, in situ intrinsic process. Option B, ex situ intrinsic process. Option C, in situ engineered process. Or option D, ex situ engineered process. I hope everyone has understood the question. Please take your time and let me know what you think is the correct answer. Post your answers in the chat box or in the comment box. I'll wait for a minute or two and then come back to this question. Okay, I think it's time to revisit this question. Oldest bioremediation system is the activated sludge system. What kind of bioremediation system is this? Is it an in situ process or an ex situ process, an intrinsic process or an engineered process? Okay, let me check the comment box for answers. Ashwini has answered option D. Okay, let us see what is the correct answer to this question. Indeed, option D is the correct answer. Activated sludge system is an example of ex situ engineered process. Well done, Ashwini. Ex situ engineered process, how? Let us understand using the same logic and concepts we learned in the earlier questions. So what is activated sludge process? Let me define it for you once again. How is the process done? Activated sludge process is used to treat wastewater in a sewage treatment plant or in a bioreactor, as we call it. First, there is a wastewater enters a sedimentation tank in which sediments or soil particles and heavier particles settle down, and then it is fed into a first clarifier. Now, in the first clarifier, the sediments is removed and the organic material dissolved water is sent into a reaction tank wherein there is addition of fermenting bacteria which dissolve, which consume the organic matter present in the slurry present in the wastewater, then sent to a secondary clarifier from which the sludge is returned back to the reaction tank, which contains now already growing microorganisms, which is called the activated sludge. Now excess sludge and raw sludge is drained out into a sludge thickener tank, and then other procedures like sand filtration follows discharge and disinfecting facility. So this is a whole schematic of how an activated sludge process of bioreactor for wastewater treatment works. The important areas are the reaction tank in which microorganism and microbes are added, which consume the biochemical and organic matter present in the water, dissolved in the matter, and this solid waste, a fraction of this is now returned back to new wastewater and treated as an activated sludge. Activated sludge system, partially treated sludge with microbes is mixed with new untreated wastewater in a treatment plant. The activated sludge system is therefore XC2 because wastewater is collected and treated in a plant, not in its natural location. Yes, this is an XC2 process. An engineered process because involves deliberate aeration, recycling of microbial sludge and controlled microbial growth. So controlled microbial growth, addition of new sludge material, activated sludge, recycling, and XC2 process because wastewater is treated and collected in a bioreactor which is not its natural location. Therefore, it is an XC2 engineered process, and option D is the correct answer. I hope everyone is satisfied and cleared with the concepts. If yes, then just give me a heads up and we will move to the next question. Very good. Coming to question number 8 of this session. 3 more question number 8, 9 and 10, and we will be done with this class. Question number 8, bioremediation 2.0 refers to the use of dashed. Option A, purified enzymes. Option B, bioengineered enzymes. Option C, bioengineered microorganisms. Option D, pure bacterial culture. Option E, groups of microorganisms. Bioremediation 2.0. Go through the question carefully, look at the options, and let me know your answer in the chat box. I will be back to this question in a minute or two. Okay, I think it's time to come back to question number 8. Let me check the chat box for answers. Ashwini has answered option C. Bioremediation 2.0 refers to the use of dashed. Let us check what is the correct answer. Okay. Correct answer I have marked is bioengineered enzymes. Okay. No, I'm sorry. This will be option C. Option C is the correct answer. Might have made a mistake here. Let me rectify it. It's not bioengineered enzymes, rather bioengineered microorganisms. Ashwini, you are correct. No need to worry. I have done a little mistake. So, bioremediation 2.0 refers to the use of bioengineered microorganisms. So, this describes advanced bioremediation techniques that use genetically engineered organisms to enhance their ability to degrade pollutants, often using tools like CRISPR-Cas9 for precision gene editing. It's like engineering your own microbes to gain enzymes that can degrade pollutants better or remove contaminants. And using gene editing techniques like CRISPR-Cas, you can do lots of genetic engineering right now. Genetic engineering allows for targeted introduction or modification of genes to improve pollutant degradation, metal accumulation, or resistance to harsh conditions, making it a more efficient and targeted approach to cleaning contaminated environments. So, this is also a truth that the bacteria or the genetically engineered organism or the bio augmented organism needs to survive in the harsh conditions in the contaminated environment to do its work properly. So, any genetic modification which is even allowing it to resist the harsh conditions, allowing it to accumulate metal or improve an enzyme for pollutant degradation is bioremediation 2.0. So, what all is implicated in bioremediation 2.0? Here is a table comparing it. Metagenomics and microbial community profiling on different levels. You can find what microbes are naturally present, what genes, enzymes they have, who interacts with whom, helps identify candidates without needing to culture everything. So, bioremediation 2.0 is acting on all these fronts. First, metagenomic and microbial community profiling, enzyme discovery and engineering, isolating enzymes that break pollutants, optimizing them, and sometimes designing new ones in microorganisms. Mixed or engineered consortia, using multiple microbial species that complement each other. One breaks down a pollutant to intermediate, another handles the intermediate, etc. For example, using computational and AI tools for predicting which enzyme can reduce a pollutant, and immobilization materials like scaffolds and hydrogels for making it reusable, and biohybrids for better availability, and tackling emerging pollutants like microplastics and pharmaceuticals. So, bioremediation 2.0 as a concept depends on genetically modified organism and theoretical approaches like computational metagenomic analysis approaches for better bioremediation. So, here is an example of genetic engineering in which the genetic engineering can be used to degrade organic compounds, heavy metals, oil and components, pesticides and radioactives. So, specific promoter reporter that is cloned into a plasmid for a desired gene which is closed, and genetic vector. This plasmid harbors the enzyme that is necessary for or that is capable of degrading multiple components of classes of pollutants. This can be transformed into a host cell which can then multiply with multiple copies of the desired genes to create genetically engineered microbes assisted for bioremediation of various pollutants. I hope it is clear to the students what is bioremediation 2.0 and how it uses genetically modified organ microbes for bioremediation. Okay, cool. Moving on to the ninth question of this session. Two more questions, question number 9 and 10 to go. So I would encourage all the participants who haven't answered, please try answering these questions. Which one of the following is included in pre-genomics molecular analysis approach? Pre-genomics era. Option A, meta barcoding analysis. Option B, metagenomic analysis. Option C, meta transcriptomic analysis. Option D, 16S rRNA based identification. I think this is one of the easy questions. If you remember the genomic and pre-genomic era, you will be able to answer this question. Which one of the following is included in pre-genomics molecular analysis approach? Okay, I think it's time to revisit this question. Which one of the following is included in pre-genomics molecular analysis approach? What are the answers in the comment box? Ashwini says option D. Okay, so correct answer to this question is option D. 16S rRNA based identification is a pre-genomic molecular analysis approach. Correct answer, Ashwini. So, understanding the pre-genomic and genomic era, the history of DNA sequencing. The DNA structure was first solved in 1950s by Rosalind Franklin, James Watson and Francis Crick. Morris Wilkins all are to be credited for the DNA structure, and DNA sequencing was introduced by Sanger, commonly known as Sanger sequencing, in 1965. Now this is the pre-genomics era. Then comes the first gen sequencing, where Sanger refines the method for DNA sequencing, and Applied Biosystems commercialized the Sanger sequencers in 1970s to 1980s. Then comes second gen sequencing like pyro sequencing and Illumina from 1990s to 2012, and the current era is the third generation sequencing of nanopore and PacBio commercialized sequencers. Okay, so this is the time that we are talking about, 1915 to 1960s. The pre-genomic means method used before the era of high throughput sequencing and whole genome analysis. So whole genome metagenomic analysis is not possible, which we also encountered in another question. These approaches mainly relied on single gene markers like the 16S rRNA in bacteria for identifying organisms rather than analyzing the entire genome or the metagenome. So there was no such metagenomic analysis present back in 1960s. Meta barcoding analysis used DNA barcodes like 16S ITS combined with next gen sequencing It allows for community profiling of many species in parallel. You can tell that what are the species and which community they belong to in parallel. But this is obviously after the pre-genomic era, in the next generation sequencing era. Metagenomic analysis involves sequencing all the genetic material from an environmental sample, whole metagenome analysis. Clearly a genomics era method, but provides an insight into the community composition plus the metabolic potential of the community. Transcriptomic analysis, again, sequencing all the RNA transcripts. This is also a strictly genomic post-genomics method. High throughput sequencing is required. It also tells us which genes are being actively expressed in certain environmental conditions in the samples. Therefore, the correct answer is 16S rRNA based identification. This is highly conserved and only one locus need to be sequenced in the environmental sample with variable regions for taxonomic differentiation. PCR amplify the 16S gene, then sequence and compare with the databases widely used before genomics to classify and identify microbes. So this was the method that was commonly used in the pre-genomics era before the invent of high throughput sequencing or next generation sequencing. I hope it is clear to the students. If yes, then we can move ahead to the last question, final question of this session. Question number 10. It's a long question. A bacterial strain was genetically engineered to break down xylene and showed promise in laboratory studies. After the engineered strain was added to a river contaminated with xylene containing industrial effluent, xylene levels remain unchanged. Which of the following is not a possible explanation for this observation? I hope you understood the question. A bacteria was found out, in fact, bacteria was genetically engineered to degrade xylene, but when added to a river which is contaminated with xylene, xylene levels did not go down, it remains unchanged. Which of the possible explanation, which of the options is not a possible explanation? Option A, ambient pH and temperature of that habitat may be unsuitable for the bacterium, which were designed in the lab. The resident microbial community may be resisting colonization by the externally added bacterium. Option C, there may be a lack of other nutrients required for growth and bioremediation. Option D, the genetically modified bacterium lost its xylene degradation capability due to lack of selective pressure. And option E, co-pollutants in the industrial waste may be toxic to the bacterium. I hope you understood the question. Read the options carefully. Can be a little confusing. Related to xylene degradation, a bacterial strain modified in the laboratory, added to the site, but there is no effect. Let me know what you think is the correct answer. Post your answers in the chat box and in the comment box. Okay, I think it's enough time for this question. Let us check the chat box for answers. Ashwini only has answered option D. Let us see what is the correct answer to this question. A genetically engineered bacterial strain to degrade xylene when added to the natural in situ environment does not show any effect. And which of these is not a possible explanation? Correct answer is option D. The genetically modified bacterium lost its xylene degradation capability due to lack of selective pressure. This is not a possible explanation because xylene is still present in the industrial effluent in the in situ site. So there is no lack of selective pressure. Xylene is there. This is not a possible explanation. So this whole process is called bioaugmentation, in which the bacterial strain is now genetically engineered. So bioremediation 2.0, and this bacterial strain is added into the in situ site. Bioaugmentation is the process of introducing specific microbes, often in the form of enriched cultures or genetically engineered strains, into contaminated environment to enhance its biological degradation capability. This seeding of the polluted site with specialized microbes helps break down contaminants like pesticides or hydrocarbons more efficiently than the existing microbial community. So see, cell bioaugmentation, it can be of different types. Pre-adapted bacterial strain or genetically engineered microbe, which is in our case, added onto activated soil, contaminated soil or water mix, and may form biofilms. Gene augmentation, bioaugmentation, bacterial carriers or mobile genetic elements like transposons and naked genetic material are added, which can then confer enzymatic activity code for enzymes via the genes. Or plant bioaugmentation using phytoremediation and rhizoremediation for absorption and decontamination of the in situ site. So these are different forms of bioaugmentation, but the one that we were dealing with is the cell bioaugmentation, in which genetically engineered bacteria was added onto the in situ site. Now looking at all options, a genetically engineered bacterium could degrade xylene in the lab, but when added to polluted river it did not reduce xylene levels. Ambient pH and temperature of the habitat may be unsuitable for the bacterium. This is possible because environmental stress can prevent survival or activity of the bacterium, right? Like the wrong pH or the temperature. Because whatever temperature or pH was used in the lab to grow the bacterium may not be the same in the water. That is why it is not able to thrive there. The resident microbial community may be resisting colonization by the externally added bacterium. This is also possible. Native microbes often offer competition and can outcompete or inhibit introduced strains like predation, biofilm exclusion. There may be lack of other nutrients required for growth and bioremediation. This is also a possible reason. Even if xylene is present, microbes need nitrogen, phosphorus for biomass growth. Lack of nutrients halts degradation. And co-pollutants in the industrial waste may be toxic. This is also possible because other effluents from the industry might be killing or inhibiting the engineered strain, like toxic heavy metals or solvents. So the only possible reason is option D, which is not possible in this scenario. The river has xylene present, which is the selective pressure, and loss of function happens only if pollutant is absent for long periods. Since xylene is abundant, this explanation does not fit into the question. Okay, so this is the correct answer, option D. So with this, we have come to the end of our presentation. We have solved 10 example questions from the topics of lecture week 8. I will save this, move back to our class, stop presenting my slides. Yeah, I hope it was a fun and informative class for all of you guys, for the live attendees as well as the recorded class. So we have solved 10 questions multiple choice from lecture week 8, and I hope all the concepts are clear. If you have any doubt, you can post it in the chat box for me to clear, or if you want me to repeat any topic that you could not understand, or you want modification in the style of my teaching, then please feel free to write in the chat box, or else I will end this class. It's we have finished our questions, and see you next week Saturday at the same time 3 p.m. with lecture week 9, and a few more weeks to go till we complete lecture week 12 and finish this course. Thank you for joining this call. I hope you had a great informative session, and I will end this call now. Ashwini says thank you. You're welcome, Ashwini. My pleasure. I will end this call now, and thank you for joining. Bye bye.